We're going to talk about asexual reproduction, and this is in contrast with sexual reproduction. So sexual reproduction would be the reproduction from two different donors. You would think about humans or animals like dogs or fish, or even plants oftentimes will reproduce sexually. The opposite of that is going to be asexual reproduction. You can see the definition here: offspring whose production involves one parent. So producing offspring with one parent is going to be asexual reproduction. And there are some exceptions to this. There are some trees that are known as being self-fertile, and they can actually fertilize the pollen or the sperm. The male reproductive organ or cell can fertilize the female reproductive cell. But for the most part, when we talk about this, we're talking about asexual reproduction, one parent. So some of the benefits of this, you could call the pros or the pluses, is speed. So asexual reproduction is very quick. So thinking about maybe bacteria can reproduce multiple times in an hour. So some bacteria can reproduce, I think I've seen 20 to 30 minutes can reproduce, which is crazy fast. And it requires less energy for asexual reproduction. Some drawbacks are there's no genetic variation. So this takes a little bit of explaining. So genetic variation, look at the root words there, gene and variety, or it varies. So we have a variety pack of chips. You would expect to have multiple types of chips in that same variety pack. So genetic variation is a lot of differences in genes. A really good example for this is bananas. So the bananas that we eat, if you're talking about like the ones you just peel and eat, not plantains, but regular bananas, those are all clones of a specific banana tree. So there's no genetic variation. So if there was a disease that came around, if you own several thousand acres of a banana farm and a disease or a fungus or some kind of pest came in, it could spread from one banana to the next, and it could destroy your entire plantation, all your fields of bananas, because they are all the same. Now, if you were to switch that with if the bananas were all different types, if there was a variety of types, say 20 or 30 different, not species, but varieties there, then each of those varieties may be affected, but it's likely not going to wipe all of them out. And you can see that with any kind of plague, whether it's a bubonic plague or the Spanish flu or different examples of widespread disease around the world with humans. It doesn't ever knock us completely out. It may hit a good portion, 20, 30, 40% of the population is massive, but it's very rare when you get something that's a disease that spreads through a population that reproduces sexually, because there's so much variation, there's so much variety in their genes. So that's a drawback with asexual, because asexual reproduces and it makes offspring that are all the same. So let's talk about this. Let's recap. We've already gone through interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. We've already gone through this cell cycle multiple times. We've done it in a pie chart. We've done it with drawing each of these out. But this time we're going to kind of breeze through this and then talk about how prokaryotes go through asexual. So asexual reproduction in eukaryotes, oftentimes it looks like mitosis. And remember, mitosis is nuclear division. And then we have the three main parts of the cell cycle. And this middle part is mitosis. That's the reproductive, the asexual reproduction example. So as a review, interphase is where growth and replication happen. We see that with G1, S, and G2. And then prophase, metaphase, anaphase, telophase, PMAT. So in prophase, we see the chromosomes are forming from the chromatin. Metaphase is where we see the chromosomes lining up in the middle. We see underline metaphase, middle. Anaphase is going to be when the chromosomes are being pulled apart. The sister chromatids are being pulled apart, and it looks kind of like A's or little A's without the little bridge in between, being pulled apart. That's how I remember it's anaphase, or you can think of apart or pulled away. And then we need this typically, almost most of the time, we're going to have the same number on both sides, and that's what we need. And then we've got telophase. Think about talking to somebody on a telephone. You don't do that with somebody right beside you. You talk to somebody on the telephone that's got some distance between you. And this is the last stage of mitosis. And it is where the nucleus is actually going to divide, this is nuclear division. The nucleus actually divides, not the cell, but the nucleus divides during telophase. And the two new nuclei that have been formed, you can see that in the pictures that we've drawn, they are far from each other, telophase, and the chromosomes are starting to unwind. And then we get to cytokinesis, where the cell divides, and that looks differently. For animals, it kind of pinches off in the middle. For plants, there's a cell plate that is being formed in between. But eukaryotes reproduce through mitosis, and that's what we've spent the last 2 or 3 days talking about in different ways. Something new to you. And remember, we are eukaryotes, so our cells reproduce this way. So your muscle cells, when you tear them, your skin cells, if you get a cut or an abrasion, our cells, almost all of our cells, go through mitosis. Prokaryotes, we got to go through something called binary fission. Let's break that word down. Binary means two, so think about coding in computers. Binary means that there's zeros and ones. Bi means two. And I hope I didn't misspeak just then. The computer is broken down into zeros and ones, which is two options. There's two options there. And then fission, I think about putting Coke in a glass and it fizzing over and creating a lot of foam at the top. That is when the gas and the liquid from that drink are separating. Binary fission is when something separates into two parts, and we see that in prokaryotes. So we see the parent cell. We've got this prokaryote that's just got DNA, circular DNA around there. The DNA replicates, and I showed that with color. They're going to be an exact copy of each other for the most part, but I drew them color. They're not going to be this drastically different. I drew them this way so you could follow the DNA down. Then we've got the segregation of DNA. In history, we think of segregation as a separation of where in the 1960s is when segregation had stopped or was starting to stop, and consolidation was happening. Segregation is the separation of something. So the segregation or the separation of DNA. We've got DNA on this side, DNA on this side. This is going to look similar to what we talked about in telophase, where the DNA is actually separated from each other, and it looks similar to what we drew before. The cell splits. This would be kind of like cytokinesis, where there's two separate cells now. It's not just the DNA that's separated; it's the entire cell, and there are two identical daughter cells. So for both of these processes, we have genetic continuity. So genetic continuity is kept because we start out with a certain amount of DNA, and each of the cells that happen in this binary fission, or in this case over here mitosis and cytokinesis, the cell cycle, we end up with the same amount of DNA in each daughter cell as we had in the parent cell. And again, genetic continuity is down here at the bottom. Each new cell ends up with the same amount of DNA that it began with. So if it started out with 46 chromosomes during interphase, then as cytokinesis is finishing up, and even before then, really a little bit in telophase, each of these new nuclei, these in cytokinesis case, each of the new cells have 46 chromosomes.